next up, first of all, think about the convergence of that technology with biomedical and health informatics, or the future of virtual reality. So wide open fields out there. So next up, we have a fireside chat with two biomedical research doctors who are at the forefront of genomics, gene editing. And so Dr. John Tisdale and Dr. Bruce Conklin will share their experiences and insights on this remarkable new technology that could transform medicine and even lengthen human lifespan. Our moderator is Glenn Zoppet of the IEEE Spectrum. So with that, I'd like to welcome our next panel up to the stage.
Thanks for coming to the IEEE VICS Summit. I'm Glenn Zoppet. I'm the editor of IEEE Spectrum magazine, executive editor of IEEE Spectrum. And I have here, as mentioned, Dr. John Tisdale of the National Institutes of Health and Dr. Bruce Conklin of University of California, San Francisco School of Medicine. So we are going to talk about real science fiction stuff here today. Many of you have probably heard about genetic therapies and gene therapies and the incredible power and risk that these technologies represent and embody. And we are going to, we have two of the world's foremost experts on these technologies and this science here. And we are going to talk about a terrible and dread disease, which is sickle cell disease, which is on the verge of being cured, being fixed for the first time in human history. It's a really momentous thing that's about to happen, and we're going to find out how it's going to happen, and why it's going to happen, and what will be next after this happens. But to start, why don't we I'm just going to go right to the basics, because I know we have people with all different levels of understanding of biology, so we'll have just a little bit of Bio 101. I promise it'll be short and painless, and then we'll go into it. So we all know about genes. So basically who you are is controlled by two things, genes and your environment, nature and nurture. So the genes part is very well understood. We have, all of us, we have 23 pairs of chromosomes. These 23 pairs of chromosomes have 3 billion base pairs. And these base pairs are the building blocks of genes. There are roughly 20,000 human genes, and these genes control almost everything important about who you are. What color eyes you have, how you respond to exercise, how tall you are, and in some measure, along with environment, what diseases you're going to get and how long you're going to live. So these genes, if you took the molecule, it's a very complicated molecule, DNA. If you took that molecule out of a cell, the 23 chromosomes, if you took them out of a cell and unraveled the DNA, it would be 6 feet long. So it's an extremely complicated, extremely large molecule, one of the most complicated known. So what scientists now have is the capability to go to specific sites on a gene, and snip out pieces of the gene and put something else in it. And that capability, it has what's called germline significance, which means that it gets propagated into future cells when that cell divides. So this is incredibly important and incredibly powerful for curing disease, because many diseases have genetic basis. Sometimes the genetic basis is extremely complicated, for example, in schizophrenia and other similar maladies. But sometimes it's unbelievably simple. And in the case of sickle cell disease, one base pair out of 3 billion is wrong. So it's incredibly tempting when you have a technology like this to fix that base pair. And that's what's happening, in effect. It's not exactly that simple. Nothing is ever that simple in the real world. But in effect, that's what's going to be taking place. And we're going to talk about that now. So I'd like to get it started with John Tisdale. Can you tell us a little bit about how CRISPR works and how it's being used to combat sickle cell disease?
So CRISPR is actually a really interesting discovery in that it was an attempt to understand how bacteria resist phage infection. They sample the phage, they keep a copy of that, and then they're able to deploy these scissors really to clip that when they see it again. And we and many others thought, wow, that'd be really great if we could use that technology to help us with genome editing. So we've been trying for 20 years or more to figure out a way to correct a misspelled gene, such as the one causing sickle cell disease. But the efficiency was extraordinarily low, like 1 in a million cells you might be able to get, and that's certainly not enough to fix a disease, and certainly not sickle cell disease. But this system that bacteria use could be employed to really increase our chances of being able to do the editing, because we could take this system and deploy it as a way to cut in a very specific area in the DNA, specifically where this mutation is, and then force a recombination with the correctly spelled gene by having some DNA there to serve as a template. So I liken it to the little, the key on typewriters when we were all using typewriters. I'm pretty sure some of you in the audience used typewriters, and we're really, pleased when we got that little top left button that you could push, and it would back up and go over the letter that you had just put there, and you could change your C to an A like you meant. And so this is really a technology that allows us to do that in DNA. Just back the key up and type in the correct letter so that the right gene product is made.
So in other words, there's this never-ending war between bacteria and viruses. And in the late 1990s or early part of the 21st century, a number of researchers began noticing that bacteria had this sort of weapon that they used. They could actually, when they were in combat or invaded by viruses, they could take a snip of the virus's DNA and incorporate it into their own DNA in order to recognize and fight off that viral invader. And a number of researchers noticed that this was going on. It was mysterious at first because they saw these repeating sequences and didn't know what they were for. And then finally it occurred to some of them what they were. And then it occurred to a woman named Jennifer Doudna that you could use this as a tool to alter the gene. It wasn't the first time people thought about altering a gene using a biological tool. People had been using viruses themselves to do this, and they still do. But this one offered unusual precision and speed and kind of cleanness, if you will. So, Dr. Conklin, if you will, why don't you take up the narrative? What has happened more recently in the
Sure. So I think that I love the analogy of the typewriter and being able to correct a particular letter. The other things that we can do with CRISPR which had been done previously with other scissors, but these scissors are so much better. The other things that we can do are also to insert pieces of DNA into exact places, and then we can also cut out pieces of DNA that are there. And depending on the cell type, then different types of procedures are actually easier. One of the things that we're working on is retinal cells and neurons, the neurons which actually go to the feet and fingers, and these types of cells in particular, because one of the things about correction is that actually it's much easier to do if the cells actually will divide. And the cells that we're working with don't divide, and so the thing that is really much more efficient for us, at least, is to work on the excision, to be able to cut something out. And it turns out that there's lots of times where you have genes which are mutations where it's just the disease mutation, which the disease gene is causing problems, but if you could get rid of that, the normal gene, because you have 2 copies of each, the normal gene will support completely normal function. And so what we're focusing on is a different approach, but analogous in the sense of trying to correct the disease, but here just by removing the troublemaker, as it were, and leaving the healthy gene there. And I should say that there's over 7,000 genetic diseases, and perhaps over 300 million people, they say, have genetic disease of some sort that's significant. And most of those, the sickle cell is extraordinary in the sense that it affects so many people, and it's as a single mutation because of its history. But many of these diseases are very, very rare, and we're focusing on the diseases which will have where we can measure the effect. So in other words, if we do the experiment, you can measure the effect and tell whether or not something happened. And that's, as you know, as engineers, that's incredibly important to have that system. But often the diseases that have the most measurable effects are not the most common ones. But these rare diseases, all these rare diseases, give us an opportunity, because even just a few people, if you can measure the effect, then you can you sort of move the field forward. And those are the ones that we're primarily focusing on.
So we'll get into the nitty-gritty now. So what happens with sickle cell disease is that this one base pair out of 3 billion is wrong. And what that causes to happen is that the hemoglobin, the red blood cells, are the wrong shape, which is why it's called sickle cell disease. The red blood cells, instead of having this disc shape, they're shaped like a sickle. And what this means is that they tend to clump and they tend to abrade blood vessel walls. The result, it can be incredible pain for the people who suffer from this disease. And it shortens their life because red blood cells carry oxygen to the muscles in the body, and they can't do their job well. So what happens, you might think that, well, we've got this tool, right? And we just fix that one erroneous base out of these 3 billion. But that's not the way it happens. It turns out, for a variety of reasons, which they can explain, but I can't. You can't really do that. It's not the best way, although people are trying to do that. So what you wind up doing is something else that's very, very clever. We all have in our bodies something, we don't have it now, but we were born with something in our bodies called fetal hemoglobin, which is different from regular hemoglobin, but it works very well. Changes happen in our body shortly after birth which end the production of this fetal hemoglobin. But using CRISPR, they can actually restart the production of fetal hemoglobin. And there are 6 major big pharma companies right now racing to be the first to succeed with this. The clinical trials, or the clinical work that's been done so far is unbelievably favorable and make it fairly clear that this is going to happen soon. So do you want to pick up the discussion?
Sure. So I know I'm talking to a bunch of engineers, and you can understand that the easiest thing to do is to break things, right? So we can use CRISPR to break things, and we can use CRISPR to fix things. Fixing is a little bit harder. That's the backspace key that I was talking about. But the breaking things with CRISPR is extraordinarily easy. You know, to make viral vectors that we use to transfer genes, I need a postdoc in the lab 2 or 3 years before they're competent to do that. To use CRISPR to break things, I can take a student who's, you know, just brand new in the lab, and they can be breaking things really well by a couple of weeks in. So the concept that we're trying to develop first in sickle cell disease is to recapitulate what we've already seen happen in nature. There are people who don't switch off fetal hemoglobin because they have genetic mutations in genes that switch off fetal hemoglobin. So when they're born, they just keep their fetal hemoglobin for life. They don't switch to adult. And if they also co-inherit sickle cell disease, they never get the symptoms of sickle cell disease because the gene that turns on after birth, the gene that's misspelled in sickle cell disease, doesn't fully get turned on. They're mostly running off of this fetal. And if we could figure out which genes were broken in them to cause this, then we could just go break those genes. And it turns out there's one gene that was discovered some time ago, BCL11A, that turns off fetal hemoglobin when we're born. So that's a gene we can go try to break. So as I said, new student in the lab, I can give them the reagents to go break BCL11A and then grow out red cells from patients with sickle cell disease and see if they make the sickle hemoglobin or the fetal hemoglobin, and it'll go upwards of 90% fetal hemoglobin when we break that gene. So that's really where these companies are focused at the moment, to use CRISPR's breaking power to break a gene and cause fetal hemoglobin to go back up as it was in utero and right at birth, and to render these patients symptom-free.
So, Dr. Conklin, can you tell us a little bit about the trials that are going on in this area, the different companies involved?
Well, I'm not directly involved with them, but I think that this is certainly the area that many companies are focusing on, because you have potentially what's particularly advantageous about the breaking BCL11A example is that it's essentially like you're bringing out a spare tire, as it were, and this spare tire that you're not using now comes into play, and it fixes not just sickle cell, but all the different mutations in that same gene, so what they call the thalassemia. So there's many other mutations that occur in all over the world, and so you have this one spare tire that could fix all of them. I think one of the other exciting things is that all throughout your body with different genes that you have specialized tissues like the eye and other things, we have the same problem where there's a broken gene, and then there's also spare tires, and there's spare tires for lots of other genes as well. And one of the things that we're looking into is, say, for instance, in the motor neuron, which is the ones that I talked about that are necessary for walking, or some for vision even, some of the genes that we don't normally use in the eye, if we could bring those out as a spare tire, taking on the same analogy, you could potentially do the same thing. And essentially, it's possible because these genes that aren't on in these tissues are really held off by things that you could then break once again and bring into play. So there's really a lot of excitement, not only about the use of the BCL11A approach, which is really remarkable, and really so many people are focusing on the same thing, but also if you look at all these other diseases as well, there may also be sort of similar spare tires that we didn't even think about.
It's kind of a miraculous thing, because we find that the body has these built-in things that just aren't being used.
Right.
And in some cases, it's not even clear what they're for, but researchers have realized that these can be used to treat and cure dread diseases in remarkable ways.
Well, actually, I think, to be honest with you, researchers have not realized that. I mean, so this is really a very new concept, so a lot of things that I'm talking about this way, for instance, we're talking about bringing out spare tires that nobody's ever studied, in fact. So it's but the furthest to advance this idea is essentially the spare tire for hemoglobin, which is really a remarkable advance.
And I think there are a lot of reasons why sickle cell is now the focus. I mean, I think it's about time we did something for sickle cell disease. It's the first disease that was characterized at the molecular level. It's been more than a half a century now by Linus Pauling. We've known the genetic defect for more than half a century, yet we really haven't had any therapies that have derived from that. But there are practical reasons why sickle cell is now the focus. One of them is that, unlike the diseases that you're working on, this organ is accessible. So we can, stick a needle in the hip, draw out some bone marrow cells and take them to the lab and go do experiments. We can even get bone marrow cells into the peripheral blood now by just giving a medication and then getting it from the vein. So we have, almost an endless supply of the organ itself, the bone marrow and the stem cells from that organ that make blood, that we can do these experiments on. So we have the accessible tissue that we can modify. And then we already have experience doing transplants where we give cells to patients, bone marrow cells, to patients that have sickle cell disease as a genetic therapy. For example, if someone has a match in the family, we can do a standard bone marrow transplant. We have to modify it in a way to make it safe in this dreadful disease. But we can do a standard bone marrow transplant, give the whole genome from somebody else and all the cells that make their blood, and cure the disease. And we also know that we don't have to fix all of the cells, because when those cells are restored and they have a normal half-life of about 120 days, you only need about 20% correction to outcompete the sickle cells and have a cure. So we don't have to get 100%. We can get the tissue, we can modify the tissue, we can make the tissue divide, which makes it easier to make these techniques work and give it back. So this is these are another set of reasons why this is a current focus.
And it also points to a potential difficulty for the future, a challenge for the future, which is that in order to make this treatment work, you have to harvest some of the patient's bone marrow. The bone marrow has to come out. It has to be treated with this genetic treatment using CRISPR, and then put back in the patient. And that makes it a fairly complicated operation. And it also makes it difficult to carry out on a large scale in Africa, where a huge percentage of the world's cases of sickle cell exist. So what are some of the things that can be done to meet this challenge of bringing it to Africa?
Right, so that is the big problem that we'll face next. I mean, we can make this work in the first world. I have confidence finally. I've been doing this over 20 years, and I can say now confidently that we will make it work here. But getting it to the place where sickle cell disease is most common is going to be very difficult. Putting these cells back sounds trivial, but to do this, we really have to get rid of their remaining bone marrow, that which we didn't take out and correct, so we don't have to compete against that with the small fraction that we're able to take out. So we essentially, have to give them poison that kills their bone marrow, that's chemotherapy, and then give them this bone marrow back that we've modified, and then support them in the hospital during the time that it takes for those cells to recover. That could be a month-long process. You're susceptible to infections. You need blood transfusions during that. So you need a really highly functioning medical center to be able to carry that out with blood transfusion support. And that's not available in most places where sickle cell disease is. I mean, there are about 100,000 people with sickle cell disease in the US. There are millions worldwide, mostly concentrated in Africa. The numbers aren't known because there's no newborn screening, but the estimates from the World Health Organization are about 300,000 births per year in Africa, so many millions in Africa. So to make this work in Africa, we either need to make bone marrow transplants easier to do without all the attendant toxicity that you get from poisoning patients to make their bone marrow go away, or figure out a way to deliver these tools to the bone marrow through an IV, for example. Make some nanoparticle that'll go to bone marrow stem cells and carry the CRISPR machinery with it and do the edit in the bone marrow itself.
Dr. Conklin, you alluded earlier to other diseases that would be amenable to treatment with CRISPR. Can you elaborate on that a bit?
Yeah, well, just to comment on, I think the first point as well is that that these things are expensive. What was just talked about are expensive, but the reality is, what's CRISPR done to that? It's actually lowered the cost. It hasn't increased the cost necessarily, right? So the previous scissors that were the predominant ones were much more difficult to make, and so, for instance, although I think that the problem is real and important, that what's really going on is now it's lowering the barrier and making things much cheaper. Now, the other thing that I want to touch on, and this is something that we think a lot about, Jennifer Doudna has an institute here locally called the Innovative Genomics Institute, and which I work closely with, and she thinks a lot about how to make these sustainable therapies. And so one of the reasons actually that we're working, for instance, with the eye, and in particular, is that it's because if we can actually get the delivery methods using just the protein itself, and the protein and the guide RNA that guides it to its places, the cost actually becomes actually quite low, because you don't have to make a virus. Actually, making a virus that you can give into a person is quite expensive. But once you actually start dealing with a recombinant protein, which you can buy from a commercial vendor, and DNA or RNA, which you can actually get directly from a, essentially right off the machine, essentially, that things become, and then some chemicals, the nanoparticles, which they become very, very low cost. And so if we start thinking about scaling this up, it starts to be possible that we can think about under $1,000 per dose per patient in terms of the cost of goods going into the patient, particularly into the retina. And then if you start thinking about the cost of someone being blind, or and then, I mean, I'm not counting the physician costs and the care costs and so on, but if you could get the cost down to that level, there'd really be very little barrier to treatment. And there are, this is not as widespread, certainly some of these genetic diseases, but there are hot spots for certain genetic diseases. So certain parts of the world, there are islands, for instance, that have where a third of the people are blind for a particular genetic disease. And so one could imagine essentially, an aid program just, fixing that. And then, so it's not, the reality is that although these things are expensive, what CRISPR has done to this is actually lower the cost and allows us to think about this. And there are scenarios where it's not happening tomorrow, but there are scenarios where we can think about it being much less expensive than what it costs right now, because the cost of caring for these patients is also tremendous.
The blindness you were referring to, that's a specific form of blindness.
There's over 200 genes that cause blindness, and about 20% of them, for instance, have this particular mechanism where it's only the bad player is causing the problem. And so there's different approaches to different ones.
It's a small number of bases.
Yeah, so those are, and so there's different approaches to different ones, but there's a lot of enthusiasm around the eye in particular because it's accessible. You can actually see the cells. The cost of therapy it's an outpatient therapy to actually inject into the eye. The risk of cancer is very small because you can actually look into the eye and see if there's a cancer there, and you can and treat it, essentially one way or the other. So, the first human gene therapy approved by the FDA was an eye gene therapy. There's
just recently.
Just recently, and we think, I think this is going to be a tremendous area to work in because there really are no cures. There's things that slow things down, but very few cures that are lifelong. When we talk about editing, we're not just talking about a temporary type of cure. We're really, in that cell, it's completely cured for that, for the life of that person. So to go into a world where there's no cure for many of these genetic diseases, in the eye in particular, and people are very motivated. There's no way out. There's no eye transplant, for instance, that they can go to. So as an area, I'm actually trained as an internist, so I'm not an ophthalmologist myself, but I'm coming to this as a geneticist, and I'm drawn to this area because this is sort of the happy hunting ground for us, right? I mean, these is where we can actually see experiments happen. We can try lots of different things, and we can measure results. And so it's a very exciting time for all of us. But it's a very exciting time that way.
So there's a big difference between the regulation in the United States and the regulation in, for example, China. There was a recent news account that a couple months ago that suggested that 86 people in China have had their genes edited already with CRISPR. That hasn't happened in the United States. But it may very well happen soon. So there's an enormous difference in the regulatory regime. This has led some folks to suggest that the U.S. is going to lose their preeminence in this technology. And there are others who have suggested that no, the problem is that China's moving too quickly, because you've both been alluding to dangers such as cancer, unintended possible consequences. Could you both comment on that?
Well, I'd say the regulatory environment in the U.S., I think, is not restrictive in our developing this technology. But there's two different places where editing is being considered. One are germline cells, right, and the other are somatic cells. Somatic cells are, adult-type cells that, like bone marrow stem cells. I don't think anyone would have any ethical issue, and there wouldn't be anything more than the standard regulatory requirements for editing a bone marrow stem cell or editing a cell in the eye, because this is not going to be passed on to future generations.
Right.
Right? But then there's also the possibility that one could edit the gene in a fertilized embryo, right? And this has also been done in China, where you could envision, let's say a couple is at risk for having a child with sickle cell disease. They do in vitro fertilization. They check the fertilized embryos, and they all have sickle cell disease. And they don't want to have a child with sickle cell disease, so they want to use CRISPR to cut and repair the beta globin gene and then implant that fertilized. So that's another place that sounds like science fiction, but is actually happening in animal models now. And this tool has allowed us to make animal models of various diseases much more rapidly than before. But it also opens the door to this kind of therapy, changing the genome that will then subsequently get passed on, that we're not sure about off-target effects. So we might fix the sickle cell gene, but we might alter some other gene unintentionally that then gets introduced into the human population. So that's a completely different animal, I think, to think about regulatory-wise.
The genome is large, for example. There's 3 billion base pairs.
Exactly.
It's difficult to know that there's been no off-target effects when you're talking about a molecule that big, or a series of molecules that big.
Right, and the tools that we have to measure off-target effects aren't perfect. So they involve amplifying DNA, which has its errors that just happen through the process. So you don't know if something that you're measuring is an error of the amplification process or an error that you made from your CRISPR process. And generally, the error rates of CRISPR have been lower than what we can detect by these methods because they're lower than the error rates of the detection method. So we're not really sure that we're not doing something that we don't want to do.
Dr. Conklin, did you have anything to add?
Yeah, no, I think that the off-target issue is, I was actually a naysayer when this I thought people were rushing to therapy actually too soon, and in about 4 years ago. But all the targets that I said should be hit have been hit in terms of figuring out off-target effects for editing a cell which is not going into the germline. So in the eye or in other tissues. I'm fairly confident that we can do that safely. Some tissues are probably safer than other tissues, but still, I think we can do that. But when we talk about going into the germline, we're not just talking about one generation, but the generations afterwards. And
all of them.
And taking a responsibility for that is something that people are rightfully very hesitant about. However, will it happen? Of course it will. It probably will happen in the next 10 years in terms of for some things, and I think that it's probably fairly likely that it will. But at the same time, it's probably going to, the indications are very, very small. For the vast majority, 99.9% of all genetic diseases probably could be easily screened with pre-implantation genetic testing and things like that. So that there's actually the use of it in terms of medical terms are going to be very, very small, I believe. And also the other issue is when we talk about germline editing, passing to the next generation, it's already happened. And it's already happened when people did the mitochondrial transfer into eggs to cure mitochondrial disease. That's already happened. It was approved in England, and there's children that are already born, and their generations for generations will be carrying the mitochondria. So it's not using CRISPR, but another technology. So changing the germline has already happened, and it's gone under regulatory methods which we approve of, or most people would approve of, and I think it can happen, but it's a very, very small segment of what's necessary.
So we have time for some questions, if there are any. Yes, sir.
So we're talking about just another technology here, and we are all engineers, and we have developed a lot of technologies. I know that when the transistor was invented at Bell Labs in 1950, it transformed the life of every man, woman, and child on this earth. But to me, the technology we're talking about here is much more pervasive, much more impactful, much more dangerous in some ways. And so my question to you is, that what type of regulatory environment do the scientists work in? Put in this, we have a Society for Social Implications of Technology, and this is another technology. And we're talking about scientists around the globe who will have access to this technology. And you talk about germline transferring of genes and so on and so forth. And one could make a Pixar movie about eliminating the whole human race, right? So my question to you guys is, and I'm not putting you on the spot, what is your regulatory mechanism? Is just the gentleman behavior of a scientist, or do you have something to control yourself, or what, so I can sleep better tonight?
So we have a regulatory environment that's already been hashed out with what's called gene therapy. So we've been working for the last 20 years to develop and engineer viruses to transfer genes into cells. And that actually we have in the clinic now, we're doing in patients. And maybe surprising to you, the most popular viral system for delivering genes to bone marrow stem cells to treat blood diseases, including sickle cell disease, is HIV. So we've cut HIV up into bits, gotten rid of the infectious parts, put the beta globin gene in, correctly spelled, along with the bells and whistles that make it turn on at the right time. And we can use that as a Trojan horse to put that gene into bone marrow stem cells and then do a transplant. And that's currently in the clinic. That's regulated by a couple of entities, including the FDA. So these protocols go through the FDA and get vetted. There's a recombinant DNA advisory committee that's been around for 20 years now that reviews these kinds of interventions as well. And
Hospitals have their own review boards.
then we have review boards in the hospital. So we have our own scientific review board. We have our own biosafety review board. Then we have an institutional review board that reviews the ethics. And then once you've passed through all of those 6 or 7 regulatory bodies, you are able to move forward into humans.
And I should say also, back in December 2015, there was already Jennifer Doudna and several of her colleagues actually recognized that this was something that everyone needed to examine and think about, especially for the germline editing, but for all other things. And I attended that conference, and what was remarkable was it was the leaders of the Chinese Academy of Sciences, the European bodies, and we all went in thinking that something new needed to happen. And after all everything was discussed, there was really unanimous understanding that actually that the regulations that are currently in place are adequate. It's just the question of whether they're enforced or not. So some of these hospitals that you talk about where things are going on, there's rules in place, but they're not being enforced. But that's a separate issue. The rules that regulate the new drugs, actually in most of the major industrialized world are really relatively common and probably adequate for safety, at least at this point. It's just when they're not followed, that's the issue.
Okay. Yes, sir.
Very fascinating. You describe how you kill the bad bone marrow and you replace it with good bone marrow. Now, does the bad stuff ever come back? So you have to do it again?
Usually not. But there's sometimes some cells that survive even our best attempts at getting rid of them. And so that's the battle that we have with the cells that we infuse. We've also tried to engineer ways to give those new cells an advantage, make them more sensitive to the hormones that drive bone marrow. So we have lots of other tricks up our sleeve to overcome that the bad bone marrow could come back.
So, Gene, I hate to do this. We're going to have to move on. And I would hope that we could carry on the rich discussion at lunch with some of the other people that have questions. This deserves a summit of its own. And I want to congratulate you on the work that you're bringing forward. I have a lot of questions myself, as you can see here. So if we can, maybe we could have a nice discussion at lunch. I'm going to have to get on with letting people break for lunch so we can get back here at 1:30.
Okay.
So I want to thank this panel for their contribution to science. Thank you, Bruce and Todd. Thank you very much.
Thank you very much.
I wish we could just go on and on. Thank you. Thank you.